Good evening, Ishika. How are you doing? Okay. So yesterday we had discussed about various things like why did Mendel choose Pisum, sativum or the pea plant, and why did Mendel choose specifically the first monohybrid cross, and then today we'll be talking about the dihybrid cross also. What is the main difference between an allele and a gene? Why do you need homologous chromosomes as a unit in order to define an allele? And what is the difference between? Yeah, yeah, sure, sure. You can send me. Okay, you can send it to my number. You have my number. You can send your PDFs and whatever problems you have, you can send it to me so that I can help you out. If you need any help, you can always consult me. Okay, and I can have a look at which level they are asking the questions. Okay, so today we'll be talking about some of the specificities of genetics. So we had talked about the phenotypic ratio. Also, we had talked about the genotypic ratio, right? So today we'll be talking about the dihybrid cross, how to find out phenotypic ratio, genotypic ratio there, and also we'll be talking about the test cross and the back crosses also. Okay. So first of all, I'll briefly tell you what a test cross is. Okay. It's a very simple thing in genetics, test cross. So coming back to the test cross here. So why is a test cross done? So test cross involves mating an unknown genotype individual or an individual with having an unknown genotype, an individual possessing an unknown genotype okay, can be mated with an individual with a known homozygous recessive with an individual having a known homozygous recessive genotype. Okay, so that is why test cross is done in order to find out what is the unknown genotype here. Okay, so testing for unknown dominant phenotype. Okay, you can test it very easily. Okay, so test cross, the application of a test cross is it can be used to determine whether a dominant phenotype is homozygous or heterozygous in nature, like we were discussing yesterday. Suppose I am tall, so after looking at me, you cannot tell me that I have this genotype or this genotype. Right? In order to discern, in order to know which genotype do I possess, because phenotypically I'm tall, but which genotype do I possess, you got to do, or you got to go for a test cross. Okay, so it's pretty easy here. The application is quite promising and it's really easy. Suppose I am having homozygous dominant B, okay, BB, let's take it as BB, okay, and suppose I am crossed with, so B stands for, suppose, black, capital B is black, okay, dominant small b is white, okay, let's take color here, it is crossed with, mated with this one. All four in this case will be what? They'll all four in this case will be black only. Okay, all four black means 100% will be phenotypically black. If all the individuals are phenotypically black, that way I can discern that, I can come to the conclusion that the individual that I was considering, that I was checking for, contains homozygous dominant trait capital B, capital B. Okay, because this is the known one, this is the unknown. I'm trying to find out the genotype here. Next, if I check, suppose the individual has capital B, small b heterozygous condition, I am mating him or her with, again, an individual with homozygous recessive trait for that specific trait. Okay, then what will I get here is, I'll get capital B, small b, small b, small b, capital B, small b, small b, small b. So 50% I'm getting phenotype which is black, 50% I'm getting white. If I am getting phenotypes like this, what does it mean? That means that the individual that I am trying to discern, that specific trait that I am considering, I am trying to discern whether that trait is homozygous or heterozygous in that specific individual, homozygous dominant or heterozygous, means it will turn out to be in the heterozygous state. If after crossing with the recessive one, homozygous recessive one, 50% of it shows the characters which is pertaining to, or which is possessed by the homozygous recessive trait. Okay, this one, white. That is the main example or main application of a test cross. Okay, so now if we learn about back cross, okay, so I have received the question. Okay, I'll just check it now only. Just hold on a minute. Okay, we have written the answers also. Okay. So download the first one also. Download the second image. Okay, first one is I have also downloaded. Yeah, you have been given this phylogenetic tree, which I had showed you. So did you have any problem, Ishika, in these questions? Any of the questions, did you have any problem? I don't think you had any problem because these are all very basic questions. If you had any problem, then you can tell me. If in any question you faced any kind of problem while solving the questions that you have sent me. Only the last question. Last one was, describe one driving force of evolution that isn't natural selection. This one, right? Explain what occurs and how it shapes the resulting population. Okay, describe one driving force of evolution that is not natural selection. So just think about it, what it could be. Just think. Try to think which is not natural selection. So natural selection is what? So it could be mutation, right? It could be mutation, right? So there are various types of driving forces, okay, in evolution. Okay, so many, yeah, you have written correctly. You have written the answer correctly. I just checked it. It's mutation only. As per me, is also a mutation only. Random change in DNA. Yeah, that is correct answer only. That I read just now. That is the correct answer. So in many researches also, yeah, that answer is completely correct. Deepika. Mutation is the correct answer. Okay, so since you know the answer, so that is a very good thing that you have been able to solve this question. So mutation is a very, you can say, tremendous pressure when it comes to mutation. So different types of mutations are there, which is there in your course, but it will come later on in the course only. You have transversions, you have transitions, and then you have point mutations, you have insertions, deletions, frame shift mutations, then chromosomal aberrations. So many types of mutations can happen. They are the random events. Doesn't matter if your natural selection is going on or not. These are independent events. Okay, mutations, and if they are prevalent in more than 1% of the population of that specific species, that means it is then termed as SNP, single nucleotide polymorphism. That is the difference between an SNP and a mutation. Mutation is always prevalent in less than 1% of the individuals of a population, whereas SNP is in more than 1% of the individuals in a population. Okay, so questions are nice and easy, good thing only. Now coming on to the back cross. So back crossing is also quite easy. Okay, so back crossing is a crossing of a hybrid with one of its parents. Okay, with one of its parents or an individual genetically similar to its parent. Okay, either to one of its parent. I'll just write the definition. Back crossing is a crossing of a hybrid, right, with one of its parents or an individual genetically similar to its parent in order to achieve offspring. With a genetic identity. Identity which is closer to that of the parent. Of the parent. It is used in various cases, like it is used in horticulture. Okay, it is also used in animal breeding, and also it is used in producing gene knockouts. Later on, you will be learning about gene knockouts. You knock out one of the allele or both the alleles you knock out means you make that specific gene dysfunctional, so that you can test whether you have been able to, you can say, go for that null protein in that specific, so suppose organism, suppose you don't want the insulin to be produced, so you knock out the genes of insulin, you cause some nonsense mutation. Nonsense means stop codon mutation, that I'll be teaching you later on. Okay, so thereby you can go for, you can say, gene knockout experiments also while back crossing. Okay, It's a very easy stuff. Okay, so not that you can say difficult, any kind of back cross. So if you have you can say if I make a cross here, T T is the F naught generation. This is the F naught. They become TT F1. Okay. Now F1 generation. Okay. If I do back cross means this one with this one similar trait. This is also tall. This is also tall. So similar trait I am going for. This is the difference between back and test. Test means always the homozygous recessive will be crossed with any unknown. Here similar trait. Okay, similar trait is used. Okay, so TT will be crossed with capital T. I can write it here, small t, so capital T. Capital T. So I'm crossing it with this one. And what I'll get is capital T, capital T, capital T, small t, capital T, capital T, again capital T, small t. All will be tall. All will be similar to the parent. So that is what is the application of back cross. That is why it is different to, you can say, test cross. Okay. And this is going back, so that is back crossing. Back cross. So now we will be coming on to the specialized dihybrid cross now. A dihybrid cross in Mendelism, then we'll come to the laws of Mendel, law of dominance, law of, you can say, segregation, and law of independent assortment. So let's do the dihybrid cross. Dihybrid means we are considering two traits or two characters, means two specific genes making two different proteins that we are considering here. Okay, so mostly we consider this thing RRYY, small I, small y. Capital I is the dominant, capital Y dominant, recessive, F naught generation, so it will be homozygous recessive. This is round, dominant, round seed, round and yellow, color and shape I am considering here. So round and yellow, this is wrinkled and green. So this is what I am considering. Okay. So round and yellow, wrinkled and green. So the thing is, if I go for the F1 generation, we all know that what we will get is this only. Okay, we can do two, you can say, monohybrid cross. There are ways to do it. Okay, there are various ways to do it. One is a monohybrid way, one is a dihybrid way. So we can go for two monohybrid crosses. Okay, what we'll get in the end is this only. Okay, we'll get all the individuals or all the plants, all the Pisum sativum, phenotypically they'll be round and yellow, but they'll be all be heterozygous. Okay, round yellow but heterozygous. So we can get it done by two ways. One is we can go for two monohybrid crosses. We can go for like this. So what are we getting is this only. Okay, or we can go for two dihybrid crosses. Small y. Two monohybrid cross is the second monohybrid cross. Okay, not getting for direct dihybrid cross. So what I am getting here is capital Y, small r, small y. So when I combine this, I am getting this thing. One way you can do like this, or one way you can go for directly the dihybrid cross. Still you will get this only. So this is a proof. Okay, this is a cross verification proof that you will get this only. Okay, but now arises the problem. Now you have F1. In F1 you have this specific genotype. Now you will be doing what? Selfing, crossing with the same genotype of F1 only, and then you will be thinking of getting F2. When you are pondering upon that specific problem, then it becomes quite tedious, right? If you are shooting in the air and trying to do monohybrid, dihybrid, it becomes quite tedious. So one trick is you do like this: capital R with capital Y, capital R with a small y, this R select with these two, and then this small r also select with these two, and write here. So capital R, capital Y, capital R, small y. First I did this, then small r, small y, then small I, r I combined with small y. Okay, I got these 4 specific genotypes. Repeat this 4 here. Repeat this 4 in the same chronological order here. Okay, and then we can go for crossing. Now this one will be crossed with all 4. Again this one will be crossed with all 4, means this one all 4, this one also all 4, this one also all 4, and this one also all the 4. So first one R R Y Y. Okay, then capital R, capital R, capital Y, small y, then capital R, small r, capital R, small r, capital Y, small r, capital Y, small y, then capital Y, sorry, capital R, capital Y, small y, small y, small y, capital R, small r, capital Y, small y, capital R, small y, small y. Okay, coming on to the third one. Capital R, capital Y, capital R, capital Y, small y, then small r, capital Y, then capital Y, then we have small r, capital Y, small y. Okay, then last one is small capital R, capital Y, small y, capital R, small y, small y, then small r, capital Y, small y, and only the last one will be capital R, small y, completely homozygous, only the last one. Okay, so this is the cross that you will be getting. Okay, after you do go for dihybrid cross, which becomes very easy after you apply that specific trick, that capital R with both the y's, there is small r with both the y's, you write it on this direction horizontal, you repeat it in the vertical direction, then you go for the crossing. Okay, so this is a specialized trick. I'll tell you one more trick by the theory of probability. Okay, that also we will be looking at. Okay, so this is the way it is done. Now go for the phenotypic cross. So phenotypic ratio, sorry, not cross, phenotypic ratio will come up to be 9:3:3:1. Just check. 9 will be round and yellow. Okay, 3 will be, you can say, round and green. Another 3 will be wrinkled and yellow. 1 will be wrinkled and green. So check for this one. This is first round and yellow. So for round and yellow, second, third, fourth, fifth, then this one sixth, this one seventh, this one eighth, then this one ninth. Okay, so 9 are round and yellow, you can see here. So for round and yellow, what do you need? I'll tell you the genotype for all. Okay, how do you get? In order for you to be round and yellow, what all genotypes do you need? Either you can be this or you can be heterozygous. You can be this one. You can be this one also. Or you can be this one also. So this comes to be 9 genotypes are like this. So round and yellow. Okay. Now let's make one of the characters recessive. Let's make it wrinkled yellow. if you are looking at wrinkled yellow, then wrinkled must be small r, capital Y, or small r, capital Y, small y. They will be three in number. Okay. Then you can say round and green. If I'm talking about round and green, it will be capital R, small y, or capital R, small y. They are also three in number. Then wrinkled and green, both recessive. Okay, wrinkled and green. There only one, that is small r, small y, completely homozygous recessive, one. That is 9:3:3:1. Okay, this is how you get the ratio. Okay. So any doubt in this ratio, Ishika? In this phenotypic ratio? Any doubt? How did we get it? Any queries till now? So now we can go for one more way of solving it. Okay, good. So one more way, that is the way of probability. Okay, two independent events happening. Okay, so this thing, selfing, is again I can go for two monohybrid crosses or not, right? I can go for this cross. Instead of going for that huge one, I can go for this one also. So I'll get this one. Okay, and then I can go with this one. Okay. So now when we have gone for two monohybrid crosses, okay, what is the probability, okay, of getting, you can say, small r here, that is 1/4. Here it is 1/4, right? Here also it is 1/4 for small y. So what is the probability that you get small r and you get small y together? So here we apply the multiplication law of probability. Okay, law of multiplication of probability, very easy law. You might have read also in your mathematics. So law of multiplication of probability. That means the probability that events A and B both occur is equal to the probability that event A occurs times the probability that event B occurs. Okay, given that both are independent events. Okay, both are independent events. So the thing is, I can just multiply 1/4 into 1/4. How much will I get? 1/16. And that is what I was getting there also, because if you add the ratio 9, 3, 3, 1, 9 plus 3, 12 plus 3, 15 plus 1, 16. So out of 16, only one is small r, capital Y. Here also I am getting the same thing. Okay. If I am going for this ratio, I am getting the same stuff here. You can see I am getting the same stuff here, that this is the same thing that I am getting. Okay. So probability rule can also be used to get the same value. Okay, to get the same value from the two specified monohybrid crosses. Okay, law of probability can be used very easily. Okay. So now some numericals, if you are given. Okay, if you are asked in a way that, suppose there is a question that there are 1000 pea plants in the F2, okay, generation, So what is the what is the number of you can say this one. Let's take the first one only, round and yellow. What is the number of round and yellow? So how will you get it? 9 by 16 into 1000. Round and yellow is always 9 by 16 because 9, 3, 3, 1. Add all of this 16. 9 parts is round and yellow. 3 by 16 would be again round and green. Again 3 by 16 would be wrinkled and yellow, and 1 by 16 will be round and yellow. Sorry, it will be wrinkled and green. Okay, so that is how you can get these kinds of questions also. And from the law of probability, if you try, you can get other ones also. Like this one you have. You have two monohybrid crosses. So for round and yellow, I showed you the thing. Sorry, for wrinkled and green. If I show it for round and yellow, okay. So round is how many? 3 are round, okay. And here 3 are yellow, okay. Here 3 are yellow, okay. So 3 by 4 into 3 by 4, how much you are getting? 9 by 16. 9 by 16 are round and yellow because this and this are round. This and this are, you can say, yellow. Okay, now let's consider round and green. So 3 by 4 again is round, but green is only this one. So into 1 by 4, again it will be 3 by 16. Same way, if you consider yellow and wrinkled is only 1 by 4, yellow is 3 phenotypes, 3 by 4. That will again become your 3 by 16, okay, which is the again correct answer, right? So this is how you can apply the law of probability also of multiplication direct to get the ratios. If at all you do not have time in exam to go for the long dihybrid cross process, okay, so this is the shortcut, you can say. Okay, so I told you both the methods, I explained to you both the methods how it can be done. So that is very easy. You can go for it like this one, okay, by following any of the method that you choose. None of will cause the deduction of marks. Both are correct in their own way, right? One is the longer one is the shorter method. Okay, so now coming on to the laws given by Mendel. Okay, law of dominance. So first law given by Gregor Johann Mendel is law of dominance. So law of dominance, if you say, okay, so we have already read about it, law of dominance. So in a heterozygote, the allele which masks the other is referred to as a dominant, while the allele which is being masked is referred to as the recessive. We know that. So dominant alleles are expressed where? Dominant alleles are expressed in specifically heterozygote, because homozygote there is no way that dominant will only express because both are dominant. Okay, so only in the heterozygote, whereas recessive alleles are only expressed if the organism is homozygous for the recessive allele. Okay, so that is known as the law of dominance always. That is the fundamental view of law of dominance. Okay. Now, if we talk about the definition, okay. So, if we talk about the definition, so I shall write it down for you. Law of dominance definition. That was the conceptual part. The definition is when parents with pure breed or with pure contrasting trait. Pure means true breed, homozygous dominant or homozygous recessive. That is also known as true breed, also known as pure breed. Okay, with pure contrasting traits, are crossed together only one form of trait appears in the next generation. So the hybrid offsprings will exhibit only the dominant trait in the phenotype. So this is the perfect definition of law of dominance. So I had explained to you already what is law of dominance in the last class only, that this is not a fist fight between the two alternate forms of the same genes present on the homologous chromosome at the same locus. Okay, what happens is one would contain mutation, which is another force of evolution only, except the natural selection that you have written correctly in your exam. So the thing is, mutations or SNPs, they can change the codons that I had showed you. If they change the codon, the amino acids can be changed, and when the amino acids can change, the conformation of the protein can become really horrendous and really unstable, you should be saying. So unstable conformation may not be having perfect active site, perfect binding site to its receptor, and thereby it may not be able to exert the same effect that the perfectly configured or structurally stable protein can. Okay, it can cause stop codon also formation, thereby it will form a very truncated mRNA with no protein also. That can also be the case in case of nonsense mutation. The one that I showed you yesterday is missense, that amino acids are being changed, missense mutation. I'll explain the mutation part to you in detail later on when the course comes, or in your course when the mutation is taught in your school. So that is the main, you can say, fundamental view, okay, regarding the mutations. Okay, so recessive mutation is one thing. Some can be dominant mutations also. Okay, the thing that has been explained to you is only the recessive ones. Okay, the recessive ones are repressed, but sometimes what happens is the dominant gene, suppose you have capital T, okay, so that can also cause a disease state. Okay, capital T sometimes can also cause a disease state that is known as dominant autosomal mutation. Okay, what it can do is, what can happen here is, it can have some very expressive promoter, thereby its mRNA is made all the time. Okay, and then its protein may bind to its receptor too tightly. Okay, that can also happen, that we do not think of. Okay, so Suppose I told that recessive one doesn't bind to its receptor properly. Fine, small t, HGH doesn't bind to its receptor properly. So you're getting a dwarf person. But if it is a dominant mutation, meaning that this one is different, this one binds to its receptor too tightly, always stays bound. So you may get excessive growth, okay, which is also not desirable for your body. Excessive growth, your heart may grow abruptly, your feet may grow too much, your head skull size may grow too much, your weight will be too heavy for your muscles to take, right? That is known as, you can say, dominant mutation. Okay, so these kinds of mutations are also there, okay, but it is not shown in the law of dominance, but later on it was discovered that these kinds of mutations also happen. Binding must be optimal. Okay, if too tight binding happens, that is also a great way of, or a reason behind occurrence of various diseases known as dominant autosomal diseases. Later on they will be taught. Okay, so any doubt, Ishika, in law of dominance? Here in the second part, where the dominant can also act as disease carrier, okay, which I just showed you to give you the real perspective of molecular biology. It is not always recessive which causes the disease. Okay, so then we move on to law of segregation, second law. Okay. Law of segregation. Second law. So during the formation of gamete, the definition is, during the formation of gamete, each gene separates from each other so that each gamete carries only one allele for each gene. One allele for each gene. Okay, that is the definition of law of segregation, and the application I'll tell you in meiosis part. So I'll just explain to you a little bit of meiosis. What happens is, in meiosis 1, the homologous recombinants or the homologous chromosomes, first of all the homologous chromosomes, they pair up. Okay, means that in a cell you have 46, so all the 46 will double up. Okay, all the 46 will double up. So first chromosome pair, okay, will pair with the first chromosome pair from the father. Suppose this is from the father, this come from the sperm, this will pair with the first chromosome pair which has come from the mother, because in a 46 chromosome cell, diploid cell, 23 have come from father, 23 have come from mother. So it will pair with the first chromosome set of the coming from the mother. Okay, and after that, in one of the stages known as pachytene, what happens is, okay, in zygotine they form synaptonymal complex, meaning that the centromeres would join together, attach together, and the pachytene they go for crossing over. Means they go for something like this. Okay, means the chromosomal arms are exchanged. This is a great way of inducing variation, even more than natural selection and mutation. This is also a way of generating variation. So in your answer you could have written mutation. One more answer would have been recombination or crossing over. This also generates. Okay, so you can write the third point in that question in evolution: recombination or crossing over. What is the meaning? I'll just show it to you. Okay, what is the effect of homologous recombination? Because it is happening between the homologous chromosomes. Okay, now let's say this is the these are the sister chromatids coming from the father. So I have a gene A here. So it will be duplicated exactly. So gene A, so suppose gene B, so suppose gene B. Okay. Here I have coming from the mother. Okay. Same gene, but little bit difference will be there. So I'll call it a different allele, right? Allele present on the homologous chromosomes. Okay. So that is different allele. So this is small a, this would be B. A, B, here it will be capital, again capital A, because it's a sister chromatid. So capital A, capital B, same. Okay. Now here it will be small a, small b, small a, small b. Now homologous crossing over, or you can say homologous recombination. Recombination means recombining. Re plus combination, recombining if you break the word. Or crossing over means you are crossing over. You are going to somewhere else. You are just crossing over. You just crossed over, right? So that means that always know the distal chromosomes. They never take part in crossing over. The first two chromosomes, the you can say proximal chromosomes or the chromosomes nearby only take part. So suppose A has this has crossed over to this one and this has crossed over to this one. Okay. So after that, what are you thinking of generating? After that, I'll generate this thing only. Here capital A, capital B is fine. But this A has crossed over with small b. Okay. So here it will be capital A, but here it will be, you can say, small b. Crossing over happened. In this case, what has happened is, here it is small a, okay, but here it is capital B. Okay, so they have exchanged places. You can see BB has or have exchanged places, whereas this remains the same. Small a, small b. So this one is a recombinant, this one is a recombinant, and these ones are parental types. Okay, so this is another way of generating, you can say, evolution or more and more variation, because you are recombining genes, so you are generating more variation. Okay, so this is what happens in the first stage, and after this happens, the homologous chromosomes are separated. Okay, after this happens, the homologous chromosomes are separated. Okay, now the homologous chromosomes would be separated completely like this. They are separated. Now crossing over has happened, now they will be separated. Okay, so 23 chromosomes will be in one cell after meiosis 1. Okay, but with 46 DNA, each has 2. Okay, after meiosis, then meiosis 2 will begin, and then you will divide this. Okay, sister chromatids also. Then what you will get is 4 cells with 23 chromosomes, 23 DNA. This you are getting, right? So 23 chromosome, 23 DNA. 23 chromosome, 23 DNA. So from a diploid cell, you are getting 4 haploid cells. That is what is known as reduction division. And here what we did, we just separated the allele so that each gamete gets only one allele. So each gamete is getting one allele here. So this is law of segregation happens in, you can say, anaphase 2 of meiosis. That is the significance of law of segregation, not by the definition, but via the, you can say, the mechanistic feature of meiosis. So anaphase 2 is this stage. When I will be teaching you meiosis in detail, then I'll tell you this is how it is, why it is called as the anaphase stage. But this is the stage where the law of segregation has taken place. Okay, and that is why law of segregation is put in the Mendel's or is taught to us. It is not just to know the superficial definition, just to understand the definition from the molecular biological perspective. Okay, most of the students, they just mug up the definitions. Okay, but the thing is, we should know the cell division aspect, the cell division related reasoning, the molecular biology related reasoning pertaining to all the laws given by Gregor Johann Mendel, all the 3 laws. That is known as law of segregation, law of independent assortment, and you can say law of dominance. So this is how law of segregation works. So each one will only have one allele, so that when it combines, so each one is what here? Each one is a gamete. If this is a female, then each one is an ovum. Okay, so when it will combine with a zygote, okay, so it's like in me also and in you also, these things are happening. Okay, so I am also producing, suppose 4 sperms like this. Okay, like this, a male produces 4 sperms. Each haploid means each has one allele only. Okay, for each trait, one allele only, not a set, one allele only. And the female also produces, these are the sperms, these are the ovums or eggs. Females also produce, the females also produce, it will be known as ova. Okay, ova in plural, ovum in singular. So ova, their ova will be like this, one allele only. Okay, and now by probability, when they meet up, okay, suppose this and this met, okay, to fertilize and form the zygote, okay, then they develop this thing. So now suppose this is chromosome number 1, I am talking about this is chromosome number 1. So now they have paired up and now they have alternative form of the same gene, that is 2 alleles for one trait now. That is why the segregation is done, so that we give evolution a chance to make the pairing up and then decide, and then the body decides which one is dominant, which one is recessive, depending upon the genetic sequence of that allele only. So this is again a direct consequence of evolution only. Okay, so think about how clever and efficient nature is regarding our own structure and functioning. Okay, so was everything clear, Ishika, in today's lecture? Any kind of queries? Okay, that's why I'm going slow in this chapter, genetics, so that each and everything must get inside your brain from molecular biological perspective. Okay, that's why I told 2 ways to solve the, you can say, dihybrid cross. You can use any one of them, even if your teacher has not taught you, even if you can use any of the methods. Okay, okay, good, Ishika. You have been doing well. So if you have any other worksheet that you want, you can send it to my number. Okay, anytime. Okay, don't ask me, just send it to me. Okay, I'll help you out, no worries. Okay, you can send anything at any point of time. Okay, just send it to me and I'll explain the reasoning to you. Okay then, thanks a lot, be safe and have a nice day. Thank you, let's meet in the next class.